Majority of the starch is stored in the endodermis itself. That is why endodermis in a dicot stem is also called as the starch sheath. If you take a transverse section of a dicot stem and observe it under the microscope, we can find that it is made up of different types of cells which are particularly arranged in certain places inside the particular stem. What are collenchymatous cells? Collenchymatous cells are a kind of parenchyma cells where they have suberin depositions or they have some depositions around them. Therefore, there are no intercellular spaces between them. Hello everyone, a warm welcome to the session on First PUC Biology. I am Dr. Divya, Biology Faculty, Vidyashram Pre-University College, Temple of Excellence, Mysore. So in this session, let us learn about the topic anatomy of the stem of dicot and monocot plant under chapter 6, that is anatomy of flowering plants. So we'll learn what type of cells are present inside the dicot and the monocot stem and then move on to learning about some of the MCQs under this particular topic. So to begin with, the dicotyledonous stem. So we'll start with the dicot stem. So if you take a transverse section of a dicot stem and observe it under the microscope, we can find that it is made up of different types of cells which are particularly arranged in certain places inside the particular stem. So based on where they are placed, they perform different functions. So what exactly is there or what type of cells do you find when we take a transverse section of the dicot stem? We will look into. So it has the outermost layer. So if you remember, in the first session of this chapter, we had studied about the general structure of the cells, or we had studied what type of cells or what type of tissues the plant is made up of, wherein we had studied about the three tissues, that is the epidermal tissue, the ground tissue and the vascular tissue. So same way we have to start here. So for the first thing that we observe, or towards the periphery of the section that we take of the dicot stem, we find the epidermis. So epidermis, therefore, because epi means outside and derma means layer. And the main function of the epidermis is to provide protection to the plants. And therefore, it is the outermost protective layer. And apart from that, it also has one more layer, that is, there is a layer that is present above the epidermis which is called as the cuticles. So cuticle, they are usually waxy, and one of the main function of the cuticle is, especially in the hydrophytic plants, that is those plants which live in water, the cuticle, because it is waxy, it acts like a water repellent, protecting the inner cells of the plant. So that is their function. So it consists of a thin layer of cuticle and it bears trichomes. So why am I using the word trichome here? Because in that session we had learned that there is a difference between the hair and the trichome. The thread-like structures that arises from the root, we call it as the root hairs, and those hair-like structures that arises from the epidermis of the stem, we call it as the trichome. So they bear trichomes, and apart from that, the stem also has few stomata. So all this, what is stomata and all that, you can go back to that session and have a look at it to understand that. Next, moving into the cortex. So we have finished with epidermis. So what is the epidermis made up of? It is completely parenchymatous. They are made up of barrel-shaped parenchymatous cells, and some of the cells in the epidermis, they get modified to produce the epidermal hair or the trichome in the case of stem. So much we know. And some of them have a covering around it, which is nothing but the cuticle. Next is the cortex. So cortex is always present below the epidermis. So it's placed between the epidermis and the endodermis. The cortex, here the cells are arranged in multiple layers. Sometimes the cortex can just be made up of only parenchymatous cells. In some cases, in the cortex, they may be made up of parenchymatous cells, collenchymatous cells or sclerenchymatous cells as well. But majority of the plants, they have a parenchymatous cells. The cells are arranged in multiple layers between the epidermis and the pericycle. Therefore, it constitutes the cortex. Along with the endodermis, we call it as the cortex. And the cortex is again divided into three subzones. So what are the subzones? Let's look into. So three subzones of the cortex, it is the hypodermis, the cortical layers and the endodermis. Hypodermis. So, in a dicot stem, in the cortex region, the first few layers that we see in the cortex region, it is made up of the collenchymatous cells. So, what are collenchymatous cells? Collenchymatous cells are a kind of parenchyma cells where they have suberin depositions or they have some depositions around them. Therefore, there are no intercellular spaces between them. So, the first layer that is there, can you see below the epidermis, the first few layers that are there, that is called as the hypodermis. And what is this hypodermis made up of? It is made up of collenchyma cells. Next, below the hypodermis, there is one more layer of cells which is called as the cortical layers. Okay, so I told you about the hypodermis. Now, what is the function of the hypodermis? Hypodermis actually gives mechanical strength or mechanical supports to the stem, because the stem is the one that holds the entire aerial parts of the plant. So, that is the reason. So, therefore, the hypodermal region, which is one of the main zones in the cortical region, which is present exactly below the epidermis, is made up of what type of cells? It is made up of collenchyma cells, which are nothing but parenchymatous cells with no intercellular spaces. And the main function of the collenchyma cells or the hypodermal region, or the cells present in the hypodermal region, is to give mechanical support to the plant. Next, moving to the cortical region. So, this cortical region are a few layers of cells which are present below the hypodermis. And what is it made up of? It is made up of parenchyma cells which are round in shape and they are covered by a thin wall, and they are parenchymatous. So, therefore, because they have a thin wall, they have spaces. So, this one parenchyma layer which forms the cortical layer, that is the second subzone of the cortex. Next, moving on to endodermis. So, how we learnt about epidermis, right? Epidermis is made up of parenchyma itself, but they are barrel shaped. Same way here also. Endodermis is present below the cortical layer, that is below the parenchymatous cells, which forms one layer of cell, and that layer again is also made up of parenchyma cell itself, but it is barrel shaped. So, endodermis, it is the innermost layer of the cortex. The cells of the endodermis are filled with starch grains, and therefore the layer is also called as the starch sheath. And we know that the starch is, it is stored in the form of starch. Food in plants is stored in the form of starch, wherein it then gets converted into glucose and moves to different parts of the plant body. So, therefore, because starch is, majority of the starch is stored in the endodermis itself, that is why endodermis in a dicot stem is also called as the starch sheath. So, next, moving on to the next layer, that is below the endodermis. So, we finished studying with the cortex, because cortex is divided into three zones, wherein we studied about the hypodermis, the cortical layer and the endodermis. So, moving on to the next layer, it is the pericycle. So, pericycle is present below the endodermis and it is present above the phloem. So, above the vascular bundle phloem and below the endodermis, we find the pericycle, and it is usually found in the form of lunar, semilunar patches of sclerenchyma. So, what is it made up of? It is made up of sclerenchyma cells. So, if you can see here, this is the layer of pericycle that is present, and it is present right above the phloem, if you can see here, and below the endodermis. And what type of cell it is made up of? It is made up of sclerenchyma cells. Next, talking about the medullary rays. So, between the vascular bundles, that is between the vascular bundles. So, if you can see here, this is the vascular bundle that I am talking about. So, between the vascular bundles, they have layers of parenchyma cells. Can you see here? They have layers of parenchyma cells which forms rays. Always the cell should be drawn like this. But here in this case, you can't keep on filling the cells completely, no? So that is why, like a cake piece, have you cut a cake piece, no? Like the same way here, they have drawn only a bit of the cell. So here and all, so the pericycle will continue here. So it is like this, it will continue. So here, in between each of, so here one vascular bundle will be there, here it will be there, here. So in between these vascular bundles, they have some cells which are parenchymatous in nature, and therefore they call it as the medullary rays, because it forms a ray-like structure. So that is why. So there are few layers of radially placed parenchymatous cells which are placed between the vascular bundles. This is one of the characteristic feature of the dicot stem. Next, talking about the vascular bundles. How are the vascular bundles arranged? Here, the vascular bundles are arranged in the form of ring. That is, they have a radial arrangement. And in between each vascular bundles, what we find? We find the parenchymatous cells which forms the medullary rays. So, they are arranged in the form of ring. Therefore, it is a radial arrangement. So, the ring-like arrangement of vascular bundle is one of the most characteristic feature of the dicot stem, because in monocot stem, you don't find this ring-like arrangement. And each vascular bundle, what type is the vascular bundle? It is the conjoint. Conjoint means top phloem is there, below xylem is there. If you can see here, top phloem is there and below you can find the xylem. So therefore it is conjoint, it is open type. Why is it open type? Because between the phloem and the xylem, there is a cambial ring that is present. So therefore, what type of vascular bundle it is? It is a conjoint open type vascular bundle. So we have already done this in the previous session. So you can have a look at the videos there. So it is a conjoint open type and with endark protoxylem. So what is protoxylem and metaxylem? Protoxylem is the smallest xylem that is there. It is the protoxylem and the largest one is metaxylem. Protoxylem is endark, means protoxylem is towards the inside. It is towards the pith. If the protoxylem was here, then we would have called it as exarch. But here, because the protoxylem or because the smaller xylem cells are towards the pith, we call it as endark protoxylem. And talking about the last layer which is present in the middle of the stem and it is made up of completely parenchyma cells, it is called as the pith. It consists of large number of rounded parenchyma cells with very large intercellular spaces and they form the central portion of the stem. So this is the last region of the dicot stem. And pith is large and conspicuous because dicots, the pith is entirely covered by parenchymatous cells. But in the case of monocots, the pith is inconspicuous. That is why whenever you take a grass or something and just see the center of the stem, it will be hollow because completely it is not filled with parenchyma cells. So that is the reason. So that is one of the major difference between the pith in the case of dicot stem and that of the monocot. So next, moving on to the monocotyledonous stem. So monocotyledonous stem, here also it has the epidermis which forms the outermost layer or the outermost covering, and below the epidermis is present the hypodermis. So if you remember, in dicot stem, the hypodermis was made up of what type of cells? It was made up of collenchyma cells, right? But in the case of monocot stem, the hypodermis is made up of sclerenchyma cells. And talking about rest all, it's the same itself. Now there is a difference in the vascular bundle. So we'll concentrate on the vascular bundle here. Vascular bundle. In the case of dicot, the vascular bundle, if you had seen, it was neatly arranged in a ring-like structure, right? If you want, we can just have a look at it again. So here, can you see the vascular bundles in a neat ring-like structure? They are arranged like this. And they have the xylem here, the phloem here, and in between the cambium will be present. Therefore, it was a conjoint open type vascular bundle and they're neatly arranged in a ring-like manner and they have medullary rays in between because it is made up of parenchyma cells. In between each vascular bundle, they have the parenchyma cells. Now remember this, going back to the monocot stem, what is the difference here? Here the vascular bundles are not arranged in a ring-like manner. They are scattered however they want, wherein always the smaller vascular bundles are present towards the periphery and the larger vascular bundles are present towards the center. So that is how it is arranged. There is no ring-like arrangement here. They are scattered. And each is surrounded by sclerenchyma. There, the vascular bundles, between the vascular bundles, what was present? Parenchyma cells were present in the case of dicot stem. But in the monocot stem, what it is there? Entirely, this is sclerenchyma. It is made up of sclerenchyma cells. Therefore, the hypodermis of parenchyma is made up of sclerenchyma only. And the bundle sheath, they have the bundle, that is, they have the sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. The ground tissue is made up of parenchyma cells. Surrounding the vascular bundles is the sclerenchyma cells. The hypodermis is made up of sclerenchyma. And below the hypodermis, whatever ground tissue or cortex is there, it is made up of the parenchyma cells. So this is one of the major difference between, so you can find the difference between the monocot stem and the dicot stem in terms of hypodermis. In monocot stem, the hypodermis is made up of sclerenchyma. In the case of dicot stem, the hypodermis was made up of collenchyma cells. And talking about the vascular bundle, here in monocot, the vascular bundles are scattered, wherein the smaller vascular bundles are placed towards the periphery, that is towards the epidermal end, and the larger vascular bundles are present at the center towards the pith. And talking about the cells between the vascular bundles, in the case of dicot stem, it was parenchyma cells. Here it is sclerenchyma cells. So these are the major differences. So in talking about what type of the vascular bundle is of what type, here it is conjoint closed type. Why it is conjoint closed type? Because between, again conjoint means top layer will be phloem, below that will be the xylem, and it is a closed type, meaning between the phloem and the xylem they do not have a cambium or they do not have a cambial ring. If you can see here, entirely it is phloem and here you can find the xylem. So between that there is no cambium. So therefore the type of vascular bundle that the monocot stem has is conjoint closed type vascular bundle, and here and it is scattered. The vascular bundles are scattered. And it also told you the peripheral vascular bundles are generally smaller when compared to the ones that are located at the center, and phloem parenchyma is absent because they do not have, the phloem is not surrounded by the parenchyma, but instead they have water containing cavities which are present within the vascular bundle, which you don't find in the dicot stem. So the vascular bundles in the monocot stem, they have water filled cavities between them. So this is about the monocot stem. So next, let's look into some of the MCQs under this particular topic. The hypodermis of dicot stem consists of dash cells. So where is the hypodermis present? It is present. It is one of the subzones of the cortex which forms the first subzone of the cortex, which is present exactly below the epidermis. So, and what type of cell it is made up of? In the case of, read the question properly, I am talking about the hypodermis of dicot stem. What type of cell it is made up of? It is made up of collenchyma, not sclerenchyma, because sclerenchyma is in the case of monocot stem. So here it is collenchyma. Of course, parenchyma only, but what type of parenchyma? It is collenchyma. So the option that needs to be chosen here is option B. It's not aerenchyma. Those parenchyma cells which have large intercellular spaces or air cavities, we call them as, especially air cavities, we call them as aerenchyma, and that you usually find in hydrophytic plants, the plants in living in water like lotus and all that. So option D can be eliminated. The right answer for this is option B, collenchyma. Next, dash provides mechanical support to young stem. Is it parenchyma, chlorenchyma, sclerenchyma or collenchyma? So here it is in the young stem, that is especially mechanical support to the young dicot stem is usually provided by the collenchyma. So option D is the right answer. Here it is collenchyma, because the hypodermis is made up of collenchyma and that region actually provides mechanical support. Next question. The cortical layer of dicot stem consists of dash cells. Cortical layer of dicot stem. So cortical layer is the second subzone in the cortex, right? What is type of cell it is made up of? Parenchyma, collenchyma, sclerenchyma and chlorenchyma. The answer is parenchyma. They are made up of parenchyma cells with intercellular spaces. Next question. The endodermis of dicot stem is rich in. So endodermis is the region that is present below the cortex which forms the inner covering in the stem. So what it is made up of? Is it made up of dicot stem? It is rich in starch, and that is why it is also called as starch sheath. So therefore the option B is the right answer here. Next question, which of these is the starch sheath of dicot stem? Is it epidermis? Epidermis, no, it forms the outermost covering. It has nothing to do with the storage of starch. Is it hypodermis? Hypodermis forms the region of the cortex. Endodermis, it is endodermis because endodermis is, it contains elongated or barrel-shaped parenchyma cells which is capable of storing starch. Hence, it is called as the starch sheath. Option C is the right answer here. Next question. The pericycle of dicot stem consists of patches of dash cell. So, in the dicot stem, there below the endodermis, there is a layer called as pericycle. What it is made up of? Is it parenchyma, sclerenchyma, collenchyma or arenchyma? So, it consists of patches of sclerenchyma. Option B is the right answer there. In dicot stem, medullary rays are formed by. So, what are medullary rays? Medullary rays are nothing but between the. So, in dicot stem, we know that the vascular bundles are arranged in the form of a ring, right? So, in between each of these vascular bundles, they have patches of cells, right? So, what type of cell, which is called as the medullary rays? What it is made up of? So, it is made up of tangentially placed parenchyma cells, parenchyma cells between phloem and xylem, radially placed parenchyma cells between vascular bundles. Tangentially placed parenchyma cells, no, it is nothing to do with the tangential or the radial placement. It is just that between the vascular bundles, they have the parenchyma cells. So, therefore, parenchyma cells between the vascular bundles is the right answer here. That is option D. So, next question. The vascular bundle in dicot stem is of dash type. Radial, conjoint, conjoint open, conjoint closed. The arrangement of vascular bundle is radial type. Here, but the vascular bundle dicot stem is of dash type. Is it conjoint? Of course, it is conjoint. But again here, what type of conjoint is it? Is it conjoint open or conjoint closed? We have to choose between them. So, it is conjoint open. Why? Because in dicot stem, we got to know that between the phloem and the xylem, that is in the vascular bundle, they have a patch of cambium and therefore it is called as the open type vascular bundle. Conjoint open type vascular bundle. Option C is the right answer here. Next question. Which of this is not a characteristic of vascular bundle in dicot stem? Which of these is not a characteristic of vascular bundle in dicot stem? Is it ring arrangement? Yes, this is a characteristic because the vascular bundles are arranged in the form of rings. So, this option should be eliminated. Endark protoxylem, they have an endark protoxylem, meaning the xylem that is small or the protoxylem is situated towards the inside of the stem, that is towards the pith. Therefore, they have an endark protoxylem. This option needs to be eliminated. Medullary rays between vascular bundles. Yes, they have medullary rays which are parenchymatous distributed between their vascular bundles. So, this option should be eliminated. Conjoint closed type vascular bundle. No, they do not have a conjoint closed type vascular bundle. They have a conjoint open type vascular bundle wherein between the phloem and the xylem, they have a layer which is called as the cambium. So, therefore, with this statement is not correct here. That is conjoint closed type vascular bundle is wrong regarding the vascular bundles of dicot stem. So, option D is the right answer here. So, next one. The hypodermis of monocot stem consists of. So, hypodermis of dicot stem consists of collenchyma. What about the hypodermis of monocot stem? It consists of sclerenchyma. So, it is made up of sclerenchyma cells which actually provides mechanical support. Next one, which of these is not true about the vascular bundle in monocot stem? Which statement is incorrect regarding the vascular bundle of monocot stem? That is what the question is. Conjoint closed type. Yes, they have a conjoint closed type of vascular bundle because between the phloem and the xylem, they do not have a cambial ring. Evenly shaped vascular bundles, smaller vascular bundles at periphery. Yes, of course, they have vascular bundles which are smaller towards the periphery or towards the epidermis. Water containing cavities. Yes, in between the vascular bundles or inside the vascular bundles, they have water containing cavities. So therefore, all these A, C and D are not the right answer. The right answer is option B, because they do not have evenly shaped vascular bundles, because the vascular bundles which are towards the periphery are smaller, and the vascular bundles which are situated at the center towards the pith are larger in size. So therefore, option B is the right answer here. So this was about this session wherein we learnt about the transverse section, wherein what type of cells are present inside the dicot and the monocot stem. We got to know the difference between the two and also got to know some of the MCQs under this particular topic. So I hope you understood the session well. We shall meet again in the coming session wherein we'll take up a new topic and learn the MCQs under the same chapter. So see you in the next session. Thank you.